We're going to switch over now to Tanmay Bharat, who's from here at the LMB, and he's going to talk to us about cryo-EM in bacterial cells. So thanks, Tanmay.
Okay. Well, thank you to the organizers for giving me an opportunity to speak here. It's really nice to present in methods-based meeting because I can focus on one particular project rather than give you an overview of our laboratory. But just a couple of slides. Our laboratory studies cell surfaces of prokaryotic molecules, and these molecules are important because they mediate all interactions of these prokaryotic cells with the immediate environment. The story that I'm going to tell you is about prokaryotic cell surfaces. So for the non-biologists, many prokaryotic cells, including gram-negative bacteria, gram-positive bacteria, and archaea, are surrounded by a paracrystalline array of molecules that surrounds the entire cell. And these entities are called surface layers or S layers. These surface layer proteins are probably the most abundant proteins on this planet because of their high copy numbers on prokaryotic cells. Just to illustrate that, this is, so each dot here represents an S layer protein sequence, and the distance between these dots represents the evolutionary distance as measured by BLAST. And what this analysis showed us that more than 80% of all the archaea and more than half of all bacteria possess these S layers. And this is something that we started studying many years ago. And I'm not going to go into much detail about the biology, but rather focus on one particular surface layer that we've been studying for many years, actually, and this began at the LMB many years ago. So I'm going to show you how we studied the S layer structure and biogenesis on cells in this gram-negative bacterium called Caulobacter crescentus. It's crescent-shaped. It has a very peculiar cell shape. And if you take a slice through an electron tomogram at the cell surface, you can identify the usual things you would expect on a gram-negative bacterium, which is an inner membrane, a peptidoglycan layer, an outer membrane. But in addition, this organism also has this repeating surface layer or S layer. And this is going to be the primary focus of this talk. So now this is just an electron tomogram of this Caulobacter cells. So this is this crescent-shaped cell body. The cell also has an appendage known as a stalk. And at the tip of the stalk, it secretes a polysaccharide that allows it to adhere to solid surfaces, such as this provided by the edge of this hole in an electron microscopy grid. And if you see at the cell surface, the bacterium is coated all along by this paracrystalline array, which is the S layer. So now if we take a still from the tomographic movie from the top of the cell, we can see that this S layer is arranged in a pseudo-hexagonal pattern with the subunits approximately 22 nanometers apart. From the side, we can see that the S layer is not directly attached to the outer membrane, but it's positioned roughly 18 nanometers away from the cell surface, where it's known to be bound to lipopolysaccharide molecules. The S layer is functionally organized into two parts: the discrete inner domains that give this characteristic 22 nanometer repeat, and the interconnected outer lattice that coats and provides a sheath to these cells. Now, this S layer is made up of a single protein called RsaA, and the sequence, if you do analysis of the amino acid residues, it has a non-standard amino acid composition, highly enriched in alanine and threonine residues, and there are several repeats in this protein. So we wanted to understand how this protein makes up this surface layer using structure biology methods. So first thing we did, we purified the protein and we reconstituted S layers in the test tube, here visualized using electron tomography. So just for the people who don't understand, this is a movie in which we are taking sequential slices through the volume in Z, and Z represents the path of the electron beam. So here you can see we successfully reconstituted the S layer, which shows the characteristic repeat. And we took these, we used this in vitro system to solve the structure of the S layer. So how does the protein form the lattice on cells? So we took these reconstituted S layer sheets and we build up three-dimensional crystals like these plate-like crystals. And these crystals we used for X-ray diffraction experiments to solve the X-ray structure of this S layer. So the part of the protein that crystallized wasn't the entire protein. What we found was the C-terminal part of the protein had crystallized, which actually didn't bind to lipopolysaccharide. And I'll go into more detail later. So this is the X-ray structure of the C-terminal part of the protein. It forms a right-handed beta helix, forms a rough L shape. It's a serolysine type protein. These beta strands form a kind of a sandwich. And these residues appear to be bound to these heavy metal densities that were resolved in our map. And we postulated that these constitute calcium ions. To confirm that these ions are actually calcium ions, so this is a biophysics conference, so this might be of interest to several people in the audience. We utilized a long wavelength X-ray beamline, which allows us to reach the X-ray absorption edge of calcium. So these in yellow are the putative postulated calcium ions. And using these anomalous diffraction measurements, we could actually map and confirm 17 of those 19 sites as calcium. For 2 of those sites, actually one of them we confirmed to be potassium, and the other one we have no information, but our molecular dynamic simulation suggests that it might be occupied by a magnesium ion. So this set of experiments show how this protein makes a hexamer and makes the hexagonal lattice on the surface of cells. Just to summarize, we've solved the structure of roughly 2/3 part of the entire protein, which is the C-terminal part of the protein, shown here from the N-terminus of the construct to the extreme C-terminus of the protein. We've shown that it binds to several functionally relevant calcium ions, because without calcium, there's no assembly of the lattice. And we've shown that only using this biochemical experiments that only the C-terminus has been crystallized. So how is the protein anchored, or the S layer anchored to Colobacter crescentus cells? So just for the non-biologists, most gram-negative bacteria have an abundant glycolipid in their outer membrane called lipopolysaccharide. So these lipopolysaccharide molecules have a lipid A moiety that's present in the outer membrane. This lipid A moiety is connected to core oligosaccharides, which lead to a repeating O-antigen polysaccharide that reaches out from all these all gram-negative bacteria, almost all gram-negative bacteria. And it is to this lipopolysaccharide this S layer has been shown previously to bind to. So we used a genetic mutant where we inserted a TEV cleavage site between the inner and the outer domain, and used this mutant to purify away the inner domain of the S layer protein from the outer domain using the TEV cleavage, shown here in a gel filtration curve and using SDS page analysis. So here's a reconstitution experiment with LPS. If we take just the inner domain of the protein, we get a very small domains in our cryo-EM images. And we add lipopolysaccharide that we purified from cells. We get these large aggregates. But then when we cleave the lipid A part from the lipopolysaccharide, we get these discrete single particles on the electron microscopy grid, which we were able to use for single particle cryo-EM. This is the structure of the inner domain of the RsaA protein that makes up the S layer. What we found is that the inner domain had polymerized or oligomerized on the LPS O-antigen, forming this kind of superhelical structure. And this allowed us to understand how this protein is arranged in three-dimensional space. So the chemical structure of the O-antigen was known previously, and it had a main chain composed of pyrosamine mannose with these 3-methyl glucose moieties at every 6th position. So the main chain of the O-antigen was resolved extremely well in our cryo-EM density. And binding of the protein to the LPS O-antigen appeared to be calcium dependent. So this is a native mass spectrometry experiment that we performed with Carol Robinson's group, which shows formation of the superhelical oligomer. when we deplete calcium in the reaction, this oligomer falls apart. And when we add calcium again, this oligomer can reform once more, showing that LPS binding and oligomerization is dependent on calcium. But we also wanted to confirm that using structural biology. So what we've done here is we've prepared a derivative of the single particle using the heavy metal holmium, which is known to replace calcium ions in biological specimen. So this map this is a map produced with holmium, and here at this contour levels, we can identify at least one of these locations that has been replaced by this holmium ion, showing that at least this could support binding with calcium ion. Okay, so how is the protein actually arranged along the O-antigen? That was the next question we posed. And first, we resorted to MD simulations. So if you remember, I showed you there's a main chain of the O-antigen and these branching moieties at every 6th position along the O-antigen repeat. So we performed several MD simulations with different positions of the branching sugar, and we detected 2 different modes of binding of the O-antigen, where the sugar is just displaced by 3 positions, and both were equally stable in the MD simulations. So we wanted to understand which of the 2 were the reality in our structure. So then we went back to our cryo-EM map, and instead of finding density for the branching sugar at every 6th position, we found density for the branching sugar at every 3rd position, meaning that both states are present in our cryo-EM map. So both are likely to be occurring on the cell. So we wanted to confirm that on cells. So this is a Colobacter crescentus stock of wild type Colobacter crescentus. So here is the density for the outer membrane, the inner domain of the S layer, and the outer domain of the S layer. When we take an S layer mutant, these densities for the S layer obviously disappear. But then when we add the inner domain of the protein as a reagent to the medium, we can get binding of the inner domain all along the O-antigen. And the side, reminiscent of the side views of the superhelical structure that we found, which formed on the O-antigen with the RsaA inner domain. Showing that not only can this inner domain protein bind to both types of all along the O-antigen, but can also happen on the outside, all along on the outside of the cell. So now this allows us to do a very cool experiment. So this is the same native mass spectrum that we did with Carol Robinson's laboratory. Using our reconstitution experiments, our cryo-EM structure, and this native MS, we can actually count the number of hexose moieties that are present on the O-antigen of this of Colobacter crescentus bacteria, and it's actually 42. And that's quite cool that with these biophysical and structural biology methods, we can actually do experiments at this level of detail. Okay, so I've shown you lots of in vitro data. How does the S layer arranged on cells? So we performed electron tomography and subtomogram averaging, which is a method that allows us to solve structures of macromolecules directly in cells. And using this, we could solve, so this is a 4.8 angstrom structure on cells, where we can directly dock our X-ray structure shown in red, and the cryo-EM map shown in blue. There's an unexplained density in the map, which is actually exactly the point where the lipopolysaccharide should bind from our cryo-EM density. And also we see fluctuating and flexible densities going from the structure to the outer membrane that probably are the lipopolysaccharide molecules attached to the outer membrane. So this was a couple of years ago. Now we can get much better resolution from cells. So this is very recent data that's just on bioRxiv. We can get extremely high resolution structures from cells where we can directly do model building. And this is really fitting with the theme of this conference, Next Generation Biophysics, that we can move our structure biology inquiries from the test tube directly into cells. So now you might say, what's the point of doing it in cells if you've done everything already in vitro? So when doing the cellular structure, we had a surprise that in addition to the normal LPS binding site, we detected another LPS binding site in our S layer structure, which we are still trying to figure out the physiological consequences of this second binding site. Okay, so this is just to show you the power of in situ structure biology, where we can have pictures like these and go from those pictures to an atomic structure where we can understand how a molecule is arranged inside a cell or on the surface of cell in this case. Now I would like to tell you our recent experiments on understanding how this S layer is assembled on cells. So for this, we use the tagging system where we tag the S layer protein with a SPY tag, and using a fluorescent SPY catcher protein, we could follow the biogenesis of the S layer on Caulobacter cells. I'm not going to explain this, probably most people are aware. So what we found using our experiment, so this is a pulse chase experiment where the old S layer is tagged in green and the new one in red. And what we found is that the S layer is secreted in discrete parts of the cell. And as we were doing these experiments, I have to say that other people, William Merner and Lucy Shapiro's laboratory, also reported the same thing using two-color STED methods. So now I apologize, we're just putting together the manuscript. The colors are different. So the old S layer is in magenta and the new S layer is in green. So what we found is that in dividing cells, the new S layer is preferentially inserted at mid-cell and at the cell poles. So this is a demograph of all the cells that we measured, and we take plots of the dividing cells versus the non-dividing cells. You can see that the new S layer is preferentially inserted at mid-cell, as shown by this representative image. Now, what we find is that, so this is an experiment where we treated the bacteria with a cell division inhibitor, cephalexin, and an MREB, which is a bacterial actin inhibitor, A22. So what we find, so this is a localization experiment where we can measure the correlation of the new, localization of the new and the old S layer. So this is the wild type. The old and the new S layers are anti-correlated, as one would expect. When we add a cell division inhibitor, we still find the anti-correlation, but when we add an actin inhibitor, A22, this anti-correlation is lost, which suggests that the insertion of new S layer is dependent on the MREB machinery, so the actin machinery of the bacteria. This is just some higher magnification. So here you can see that there is anti-correlation between the new and the old S layer, which is lost in these MREB, A22 treated cells. And this is just showing that the cephalexin treated cells, where the cell division is inhibited, there's almost binary anti-correlation between the old and the new S layer insertion, as shown by these plots. This is another experiment that I just want to highlight that, so this is a labeling. So we've added another label, which is for HADA, which is fluorescent. Basically, this is, I'm not going to go into detail. This is just checking where new peptidoglycan is being inserted in the cell. And what we found is that the new S layer insertion is correlated with the insertion of the new peptidoglycan. So it appears that, sorry, just to go back, just that the insertion of new S layer is related to the insertion of new membrane and the insertion of new peptidoglycan. So we wanted to understand what's actually going on at these insertion sites. So here again, apologize, the colors are different again. We performed cryo light microscopy. So these are cells that we've deposited on electron microscopy grids, plunge frozen, and then imaged in light microscopy in cryo. And we can verify that this particular cell actually has an insertion site at the cell pole. And then we can take that cell and image it again in electron tomography, shown here. And then we can perform subtomogram averaging. And what we found is that at the sites of the new S-layer insertion, there were actually lots of gaps in the S-layer lattice. And these gaps, in some cases at least, we think there are new S-layer molecules actively going inside the S-layer, which was well, maybe it shouldn't have been a surprise, but it was a surprise to us that there are so many gaps at the sites of new S-layer insertion. So this is our current hypothesis on S-layer biogenesis. There's new membrane and new peptidoglycan and new LPS molecules being synthesized at the mid cell and at the cell, at the old mid cell, which is the cell pole. And at these sites, there are gaps in the S-layer. These secreted S-layer molecules are waiting underneath the S-layer, and they just go and fill out these gaps, as shown as a new S-layer insertion. Just want to summarize quickly our studies on this S-layer. So unfolded RsaA molecules are secreted through its ABC transporter at the outer membrane. These molecules go outside, they are unfolded because they're not bound to calcium. Then they RsaA molecules can bind to calcium molecules which are present in higher concentration outside the cell compared to inside the cell. This calcium binding allows the protein to bind to the O-antigen of lipopolysaccharide. And as we know, this protein can bind to multiple sites along the LPS, so it can walk along the LPS. And this is actually shown also by William Murner's group, that it diffuses on LPS. It continues to diffuse along the LPS till it finds a gap in the S-layer lattice. It goes to the gap in the S-layer lattice where the steric hindrance provided by the capsule and the lipopolysaccharide molecules is relieved, allowing the outer membrane to assemble into a full S-layer. And these are the sites where we are trying to now understand in greater detail how this S-layer insertion is occurring by our correlated light microscopy and electron tomography experiments. I think I have 5 minutes. I can just finish with a little bit of biology. So even though we've studied the Caulobacter crucentus S-layer in great detail over several years, there's a whole world of S-layer molecules out there, and we've also done a little bit of work on them, and I want to just highlight that in a few slides. So this is an S-layer from an archaeon called Halophorax volcanii. So we've solved the structure of this S-layer. It also forms a hexagonal lattice. And this protein, so this S-layer protein is made up of an array of immunoglobulin-like domains. And these, I'll let the movie play. And these immunoglobulin-like proteins are important in forming the hexameric, dimeric, and trimeric linkages. And these S-layers coat these cells, Halophorax volcanii cells that are found in the Dead Sea. I won't go into a lot of the biology. Instead of having gaps, this S-layer is punctuated by pentameric defects, and we've solved the structure of the hexamer and the pentamer, and we've verified that this is how the S-layer is built tightly around these cells. Why I'm showing you this is that these immunoglobulin-like domain containing S-layer proteins seem to be widely conserved across haloarchaea and found in all these S-layers that we've detected using sensitive sequence homology searches. Another S-layer structure that I would like to present is something that was studied several years ago by Sexton and Baumeister, and this is an image from 40 years ago. It's from an evolutionarily deep branching bacterium called Deinococcus cardiodurans. It's an extremophilic bacterium that can survive under high doses of radiation, and it can even survive in outer space for several years. So it's extremely hardy, and part of its hyperstability is attributed to this S-layer. So we purified this S-layer, and we solved its structure. This is the structure of the S-layer protein, and it's also made of these array of immunoglobulin-like molecules that make a multi-layered structure. And these are the immunoglobulin-like domains, and these domains packed together in an interdigitated array to form this hyperstable S-layer on this extremophilic bacterium. This is just deposited on bioRxiv. So 40 years later, we've solved the structure of this S layer. Why am I showing you this? Because this is also biologically relevant, that this bacterium, which is actually a diderm bacterium, meaning that even though it stains gram-positive, it's like a gram-negative bacterium. It has these immunoglobulin-like domains. And if we, well, I put this in there, sorry, because this is biophysics. So here, to confirm that this protein is actually making up the S layer, we did focused ion beam milling of these cells and performed subtomogram averaging to confirm that actually this protein is actually the S layer protein, because there was a bit of a debate in the literature. Why this is important is that these immunoglobulin array containing domain array containing S layer proteins seem to be conserved across gram-negative, gram-positive bacteria and archaea. And if you compare one of these immunoglobulin-like domains with immunoglobulin from the human immune system, there's a striking structural similarity, which has evolutionary implications on the evolution of a modern eukaryotic cell. And I won't go into details, but I will just say that we've identified putative S layer proteins in the Asgard lineage of archaea, which is the closest relatives to eukaryotic cells. So just want to end with the fact that there's S layer proteins all around in the world that are incredibly understudied. There are common principles in their assembly and structure, and we are just scratching the surface of this. And we have these cool biophysics methods to study these. And with that, I just want to thank the people who did the work, these people with an arrow, some very generous collaborators and funders. And thank you all for your attention.
Thank you, Tanmay. Questions? We've got time for a couple of questions. I think there's a couple just in the middle there.
Hi, Tanmay. Great talk. As you look across the S layer, you see there's a lot of curvature changes in these bacterial cells across the membrane.
Yep.
Did you observe any structural rearrangements? So you mentioned one other species, they incorporate pentamers.
Yeah, so I think, I mean, I don't know if I have a slide here, but we found, so the pentamers seem to induce curvature of the S layer. So here is a slide showing that. So we solved the subtomogram averaging structure of the S layer on membranes, positively curved membranes, and the hexameric structure we solved on negative curvature. So it appears that the pentameric defects, so if you look at subtomogram averaging from a cell, the pentameric defects are present in places where the lattice is highly curved or the cell is highly curved. So the pentamers might be a mechanism for the cell to induce curvature. And I should say that these cells can mate with each other, and they form these cytoplasmic bridges where they can exchange genetic material. And of course, they're dividing, and the division needs invagination of the cell membrane. So the induction of curvature is probably maybe regulated. We don't know yet. It's a great question.
There's one behind you, and then there's just one here as well.
Yeah. So what I wanted to ask, what specifically were you labeling when you were fluorescently imaging the new S layer versus the old S layer?
So we have a genetic mutant of the S layer, which has this peptide displayed on it called the SPI tag. And it's not developed by us, it's developed by Mark Howarth, who was our colleague in Oxford. And this SPI tag can be attached, can bind to a SPI catcher protein, SPI tag and SPI catcher. And this SPI catcher protein we can tag with different colored fluorophores. So it was the SPI catcher protein binding to the SPI tag with a fluorophore.
Thank you.
Hi. Excellent exploitation of all these methods.
Sure.
How important is the influence of the electron methods on the structures you're measuring? Do you have a gauge, like radiation damage is the question, the underlying question?
Well, I think the radiation damage differs from species to species. We have studied some species which are really highly radiation sensitive, some species that are less radiation sensitive. I can tell you this species, Helophax volcanii, is more radiation sensitive than Colobacter crescentus. Whether that has any physiological meaning, I'm not clear. Yeah, and the Deinococcus radiodurans is obviously was isolated in the Chernobyl, but so it can withstand a lot of radiation. Yeah, I don't know. So
Okay.
there is radiation damage. Yeah, it's a fact of life. We have to just apply less dose when that occurs.
Okay, thanks.
Okay,
Yeah.
thank you. I think we probably need to move on. So let's thank Tanmay again. Thank you.